Hello, it is Brett here to introduce Module 5. It's taking a deeper look at cell structures and functions, in particular the plasma membrane, and also starting to get into our discussions of metabolism and energetics and enzymes. Start the screen sharing. Oh, come on. That's the thing. That's the thing I want. There we go. All right. So again, we have some cells here that are rapidly dividing, and all cells need to accomplish the functions, and they need to keep their conditions different from what's on the outside of the cell in order to stay alive. There's that idea of homeostasis from way back when in Module 1. And so now we're going to get into the discussions of how cells maintain homeostasis. So the outline for this module, there's a lot to cover, and I'm sorry about that, but we just this is where it makes the most sense to put it, and also trying to get it the same week as the lab that's happening. So the big idea is we're going to break this up into two pieces. There's going to be the membranes and transport piece, and then the energy and enzymes piece. And then the membranes and transport piece, the big questions, the big ideas, really, how does membrane structure determine function? And I'll be talking about that in the next few slides. And then the next question is, how do materials cross the plasma membrane? And the short answer is that there's a variety of ways. We can classify them as passive, meaning no additional energy is required, or active, which does require energy, usually in the form of ATP. And then within those, we have different categories of movements that we'll be talking about later. And actually, I'm not going to do the talking for those. I've got a couple guest lecture videos from the Amoeba Sisters that will talk about cell transport generally, and then osmosis in a little more detail. So that's how we're going to cover that. And then I will have a separate video lecture for the energy and enzymes chunk. And there we're just going to cover the rules of metabolism and how living organisms accelerate their biochemical reactions. And the short answer there is enzymes, but then we'll talk about some of the aspects, how enzymes work, and how environmental conditions affect it, and how enzyme activity is regulated very carefully, because cells really need to have the right materials, and they need to have the enzymes working on those materials in a very delicate balance in order to stay alive. So we aren't going to really get into we're not going to take a deep dive on all the different enzymes that are working inside of a cell, but we will kind of touch on generally how enzymes function and how that helps keep a cell alive. Okay, so to start off with, the plasma membrane, what's the structure? Well, I should also say this is also known as the cell membrane. And the technical term for this cell membrane is, let's choose a different color. Let's choose a different color here. We'll make it blue. Phospholipid bilayer. And it is made of, you guessed it, phospholipids. There we go. There we go. I can type. All right. So, so if we look at the plasma membrane, this is maybe kind of an overview. There's, an internal compartment that is separated from the extracellular environment. And what separates them is this phospholipid bilayer that constitutes most of the plasma membrane. And if we take a look at a small section of this membrane, we can see that there are these little molecules and there are 2 layers of them, hence bilayer. And they are made of phospholipids, which are really, really interesting molecules. It's basically a triglyceride. So we have our glycerol here, and we've got 2 fatty acids. This one happens to be saturated. This one's unsaturated. And then there is a phosphate group attached. Ooh, interesting. This leads to some interesting properties. This means that part of the molecule is hydrophilic. So the head that has the glycerol and the phosphate becomes hydrophilic. So it will tend to orient itself towards water. And the tails remain hydrophobic because they're just fatty acids, which are all non-polar covalent bonds between carbon and carbon or carbon and hydrogen. So those are non-polar. So in this orientation, the hydrophilic heads orient either inward towards the cytoplasm or outwards towards the extracellular fluid. And then in between, we have these hydrophobic tails that can interact and form this hydrophobic barrier so that water can't easily move across it. And anything dissolved in that water can't easily move across it. Okay, oh, I need to get rid of our rotations. All right, so you are probably familiar with these kind of dual personality molecules that are partially hydrophilic and partially hydrophobic. You actually saw an example of this in the water lab when you used dish soap to sink that paper clip. Dish soap is, they aren't quite phospholipids anymore because they don't like to use phosphate in soaps and detergents for other reasons that we'll get to later in the term. But essentially they're very, very similar. So they've got, kind of this fatty acid part and then a, usually it's sodium and I forget, it's like a sodium salt of a phospholipid basically. And they use slightly different atoms in there to make it better for the environment, but still it's essentially behaves the same way. Another example of this is mayonnaise. So you might have heard, and I might have even said that oil and water don't mix. Well, it turns out mayonnaise is just that. It is oil and water. And how do they get them to mix? Well, that actually is phospholipids. And in particular, there's a phospholipid called lecithin that is found in egg yolks, which is partially hydrophilic, partially hydrophobic. In the egg, it's meant to build new plasma membranes, but we can also take that out and use it to mix up a mixture of oil and water. And then we can make disgusting macromolecule sandwiches if we want to do that. I think that's what happened in the one of the prior guest lecture videos, if you watched that. Anyhow, so these molecules are fairly common and they have some interesting behaviors. So if we just have a very small number of them, they will tend to self-assemble into these structures called micelles, which is basically a sphere with the hydrophobic heads on the outside and the hydrophilic heads on the outside and the hydrophobic tails on the inside. If we add a few more phospholipids, we can end up with this liposome here, which is kind of like a cell, but really, really tiny, right? And then we keep expanding it, and then we can end up with an entire cell. And then if we take a section of the plasma membrane, we see this bilayer sheet. So this is how these molecules behave, and it's a natural consequence of mixing them with water. So this is really kind of an emergent property of these phospholipids, which is really, really cool. Okay, so the plasma membrane, I think I mentioned this in the prior module, that it's not just phospholipids. There's some other things embedded in it and attached to it. So we have our phospholipid bilayer, and then we might have some proteins that go all the way through and might have a little space inside. We would call these channel proteins. Sometimes we have other proteins that are fully embedded and span the bilayer, and we would call those integral proteins. Sometimes the proteins are only halfway embedded into the bilayer, and so those would be peripheral proteins. On the extracellular side of the plasma membrane, you might have glycoproteins, which are just these proteins, these integral proteins, for example, with a carbohydrate attached. Or we might have glycolipids. So it's like a phospholipid with a carbohydrate attached. And then also inside this membrane, we can find a little bit of cholesterol. So actually cholesterol is good for you, or having the right amounts of the right types of cholesterol is good for you. And it actually helps stabilize the hydrophobic part of the plasma membrane. So the cholesterol molecules are totally hydrophobic, and so they'll hang out in between and help give structure to these phospholipids and proteins. Okay, so I have this analogy. If you imagine a swimming pool filled with ping pong balls, those ping pong balls might be kind of like the hydrophilic heads of the phospholipids, and they can move any which way in the plane of that membrane, right? And then if you throw like pool toys or those like, you know, floaty rings or things like that into those might be kind of like the proteins in that plasma membrane. And they can trade places laterally, but they can't escape the membrane because of the forces that are holding them in place. So I'm not going to show this video. It's kind of neat. It's kind of fun. It's in the slideshow if you want to see it. But just, you know, I saw that and I was like, oh, that's like a you know, the fluid mosaic model. So all these molecules can trade places in the plasma membrane, but they can't escape it easily since there's water on both sides. All right. So what is there outside the plasma membrane? In animal cells, there would be the extracellular matrix. So that is a network of collagen fibers, collagen being a protein, fibronectin, which is kind of this little connector protein that connects to the integral proteins. And then there are a mixture of carbohydrates and proteins called proteoglycan, which makes it kind of sticky. And what am I trying to say here? Yeah, there's kind of a network that can attach to itself and can really trap water molecules, keep the cells hydrated pretty well, especially if it's like skin cells where it's exposed to the air and water's constantly trying to evaporate. And then on the inside, the cytoskeleton connects to these integral proteins. So it's really kind of it's not just this flimsy plasma membrane. There's some reinforcement both inside and outside. What about other types of cells? So for bacteria, they have a structure outside the plasma membrane called a cell wall. And we remember back to our picture of a typical prokaryote. It's a rather thick cell wall, and it's made up of a molecule called peptidoglycan. And so it's basically a modified protein that they use for their cell wall, or a couple of modified amino acids. In plants and algae, they use cellulose primarily, and sometimes some other polysaccharides, but we're going to say mostly cellulose to form that really thick cell wall that makes up anything made out of wood, or like the plant cells that you saw in lab. That really thick cell wall was cellulose. And then in fungi, they have a cell wall as well that is made up of a really interesting polysaccharide called chitin. It's very tough, but it's also very flexible. So fungi are quite a bit more bendy. They can take a lot of different shapes, more so than plants can. And that's because they have this cell wall made of chitin, but it's also very tough. And one really, really interesting thing is that arthropod exoskeletons are also made of chitin. So it can be very, very tough, but also very flexible. And it can be kind of cross-linked when it needs to be tougher. So this is a really interesting molecule, but we don't have time to talk about that. There's the Google and the YouTubes and stuff that you can find out more about that. All right. So that's the structure of the plasma membrane. What does the plasma membrane do? I like to think of it as kind of like the velvet rope and bouncer at an exclusive nightclub. It's there to keep, to provide some separation between the outside environment and the internal environment. So if you're at a swanky club, you don't just want just anyone getting through. You want only the right kind of people to get in. Well, okay, maybe that's terrible, but for a cell, it's very important that only the right molecules come in, and other molecules are excluded, and waste products can get out, and nutrients can get in. So, so it's kind of similar, but maybe the analogy falls apart at some level. All right, well, I am going to stop talking now because there's a couple guest lecture videos about cell transport and osmosis that are going to talk more about the specificity of the plasma membrane and how it lets different materials in and out and whether or not they use energy in those processes. So I will stop this here, and then we'll have another video a little bit later on energy and enzymes. Oh, all right. I guess I could have shown this slide, but yes. Okay, more later.